We are often willing to pay 30% more just to buy shrimp labeled wild caught. In our imagination, that label promises the taste of the ocean briny, unteamed, and pure. But in a blind tasting experiment conducted by the National Fisheries Institute, professional chefs were asked to sample two types of shrimp, one caught offshore, the other farm-raised. The result, more than half of the participants could not tell the difference. A $12 shrimp tasted identical to an $8 one. That raises an uncomfortable question. If wild and farmed taste so similar, what are we really paying for? The answer lies deep within the shrimp's delicate muscle fibers, where a chain of highly sensitive physical and chemical reactions unfolds. Reactions so precise that a deviation of just a few degrees Celsius or a few seconds during processing can erase the taste of the sea you thought you loved. Shrimp are crustaceans invertebrates. Instead of carrying an internal skeleton, they wear an external armor made of chitin. Imagine a building whose load-bearing structure is on the outside, while the inside consists only of soft, vulnerable tissue. Each time a shrimp grows, it must shed its old shell, a process called molting. In that brief window, it is exposed and fragile, like an organism not yet fully formed. This biological mechanism makes shrimp muscle tissue extraordinarily sensitive to environmental changes. Shrimp muscle fibers contain actin and myosin, the same proteins found in human muscles. They function through a sliding mechanism when calcium ions are present. These fibers latch onto each other and contract, creating firmness. But unlike vertebrates, shrimp have no internal system to regulate temperature. Every fluctuation in water temperature, salinity, or pH directly affects those protein structures. That is why the sweetness and snap you experience do not come from some pristine ocean region, but from a fragile biochemical sequence, one that can be destroyed by something as simple as improper icing. The moment a shrimp leaves the water, a biochemical clock begins to tick. Its cells remain alive for several hours, consuming oxygen and releasing intracellular digestive enzymes. If not processed quickly, these very enzymes begin to digest the shrimp from within, turning the flesh mushy and bland. Freezing was invented as a way to freeze time, but slow freezing is its most ruthless enemy. As temperature gradually drops, water inside the cells crystallizes into large ice crystals, like thousands of microscopic needles piercing cell membranes. When thawed, that water escapes, leaving behind hollowed-out muscle fibers. The texture becomes soft, dehydrated, and flavorless. By contrast, flash freezing plunges shrimp to -40 degrees Celsius within minutes. At that speed, water molecules do not have time to gather into large crystals. Instead, they form a stable microscopic lattice. The tissue structure is locked in, enzymes are halted, and flavor is preserved. So what determines good or bad shrimp is not the ocean it came from, but the speed at which the water inside its body solidifies. The taste of the sea is, in reality, shaped inside a freezer. If you have ever watched shrimp cook, you may have noticed something curious. It curls into a C shape and sometimes tightens further into an O. That is not random. It is the language of proteins responding to heat. Inside shrimp flesh, protein chains are intricately folded into a water-holding network. When exposed to heat, hydrogen bonds break. The proteins unfold and reconnect, a process called denaturation. If heat rises moderately, the network restructures in a stable way, retaining moisture within. The shrimp curls into a C, a balanced form between muscle tension and internal water content. But once the temperature exceeds about 70 degrees Celsius, protein bonds tighten excessively, squeezing water out. The structure becomes dry, elastic, and hollow. The result, the shrimp forms an O, a circle of sensory destruction. Japanese chefs follow a rule known as just curl, stop cooking the moment the shrimp begins to bend, because they understand that the C is not just a shape. It is a thermodynamic equilibrium point where proteins are sufficiently opened, yet still able to retain moisture. a thin boundary between science and culinary art. If wild shrimp are an ecological gamble, farmed shrimp are a mobile laboratory. In aquaculture, farms in Vietnam or Thailand, shrimp live in recirculating systems controlled by sensors. Salinity, pH, oxygen levels, stocking density, even the type of microalgae used as feed are programmed. The result is a product with flavor consistency resembling a semiconductor production line. You can eat shrimp in New York or Tokyo and experience nearly identical taste and texture. The difference between the two shrimp no longer lies in genetics, but in environmental control algorithms. Meanwhile, wild shrimp are shaped by dozens of variables: ocean current temperature, plankton species, even minerals in seabed sediment. These create subtle flavor variations like ecological fingerprints. But that very unpredictability makes wild shrimp difficult to commercialize. And so a paradox emerges. We claim to value natural, yet we crave consistency. The market chooses farmed shrimp because it delivers what global logistics demand: most absolute uniformity. The deliciousness of shrimp is not the exclusive privilege of the ocean, nor merely the product of a laboratory. It is a temporary equilibrium between biology and technology, between time and temperature, between nature and human intervention. What you feel on your tongue, the snap, the sweetness, the hint of brine is the result of millions of water molecules arranged correctly, at precisely the right moment. So when you choose shrimp, do not look only at labels like wild-caught or organic. Look at the process from the moment it leaves the water to the instant it touches the pan, because true flavor does not reside in origin. It resides in how humans treat nature.